In the next few minutes, we are going to discuss hemolytic anemias to understand the definition, the classification, and to understand the diagnostic work up to a child with hemolytic anemia. What happens to the hemoglobin when a red blood cell dies? The hemoglobin molecule is formed of 4 globin chains and 4 heme groups. As we said before, what happens to the hemoglobin when a red cell dies? After the red cell death, the hemoglobin leaks out, typically into the blood. Hemoglobin disassembles into globins plus 4 heme groups. The globins are broken down and they are reused for its amino acids. The heme group consists of iron and porphyrin. The iron will be utilized by the body for erythropoiesis and other uses, while the porphyrin will be oxidized to biliverdin. Much of this biliverdin gets changed to bilirubin. This figure that we are going to explain in the face-to-face session shows the formation and destruction of red cells. If you start by red cells, they are produced by the marrow. They circulate, their lifespan is 120 days, and finally they go to the reticuloendothelial system in the macrophages. The red cell gets destroyed. This is in the reticuloendothelial system. When it gets destroyed, we get the production, as we said, of the globin, converted to amino acids, the heme, which will be converted to iron, carried by the transferrin to be utilized by the body and or stored in the liver as ferritin, and the porphyrin will be oxidized to biliverdin. This biliverdin will be converted to bilirubin, circulating as an indirect bilirubin, unconjugated bilirubin, goes to the liver. In the liver, it is conjugated, so we get the conjugated bilirubin, which gets excreted into the intestine, and we get the bilirubin, which is excreted into the stools through what's called enterohepatic circulation. Some of this bilirubin is excreted by the kidney, and we get increased urine bilirubin, what we call urobilinogen. So there is increase in the stools, it is in the form of stercobilinogen, and the urine, it is in the form of urobilinogen. This is what happens under normal conditions. In pathological conditions, red cell destruction can occur either within the intravascular compartment, what we call intravascular hemolysis, or it occurs in the reticuloendothelial system, what we call extravascular hemolysis. So extravascular hemolysis means red cell destruction prematurely in the reticuloendothelial system of the liver, the spleen, and the bone marrow. The best example is in the hemoglobinopathies and in hereditary spherocytosis and in autoimmune hemolytic anemia. So what happens? That the red cells, while they pass in the reticuloendothelial system, before they complete their lifespan of almost 120 days, they get destroyed in the reticuloendothelial system. So we get an increase of the unconjugated bilirubin in the circulation, and there is increase in the bilirubin excreted in the urine, the conjugated, and we get increased stercobilinogen in the stool and urobilinogen in the urine. What about if the red cells get destroyed in the intravascular compartment? We get what we call intravascular hemolysis. This can mainly, the main example is glucose 6-phosphate dehydrogenase deficiency, which we'll explain later on. It can occur in immune conditions. It's mechanical, with drugs, with infections. And what's the end result? When red cells are destroyed in the circulation, there is a release of free hemoglobin. Under normal conditions, this free hemoglobin, we called it hemoglobinemia. Free hemoglobin in the circulation is hemoglobinemia. So this hemoglobin, free hemoglobin, usually there is a protein in the circulation produced by the liver, is called haptoglobin, and its role to bind the excess of the free hemoglobin and takes this excess of free hemoglobin to the reticuloendothelial system, so it removes the free hemoglobin from the circulation. Why? Because if there is an excess of free hemoglobin, this free hemoglobin is going to pass through the renal tubules, and it is called hemoglobinuria. Hemoglobinuria results in renal tubular injury, and the end result is renal failure. So intravascular hemolysis is very serious because of the presence of free hemoglobin in the circulation, what we call the hemoglobinemia, and this helps in the diagnosis. If you take a blood sample and put it in the tube and then you centrifugate, you get red cells below and the plasma. Above, the plasma is clear. In the presence of hemoglobinemia, the plasma is stained, close to be red. And in hemoglobinuria, urine is colored because it contains hemoglobin. So laboratory diagnosis of intravascular hemolysis will include absence haptoglobin because they are consumed, hemoglobinemia, hemoglobinuria, and increased hemosiderin because when this excess hemoglobin enters the renal tubules, it's converted to hemosiderin. The renal tubular cells, it's converted into hemosiderin. It's a very serious condition because it may result in renal failure. So what are the tests to demonstrate hemolytic process? These tests include tests for accelerated hemoglobin catabolism, increased indirect bilirubin, increased urinary urobilinogen, increased fecal urobilinogen, what we call also stercobilinogen, decreased haptoglobin, free hemoglobin is increased in intravascular hemoglobin, in intravascular hemolysis, urine increased hemoglobinuria and hemosideриnuria in intravascular hemolysis, and if you have intravascular hemolysis, the red cells, destroyed red cells in the circulation are called schistocytes. They look like that. And we may get spherocytes. It's acquired spherocytes. It's different from hereditary spherocytosis. This is number one. And we may get signs of compensated increased erythropoiesis. There is increase in the reticulocytic count. Normoblast nucleated red cells start to appear in the peripheral blood. Bone marrow reveals erythroid hyperplasia, and there is expansion of the bone marrow space and showing what's called heron end appearance in the skull. Widening erythropoiesis in the bone marrow in the skull results in widening of the diploic space in the skull between the inner and outer table, and we get what we call the heron end appearance on plain X-ray of the skull. This table I will explain in the face-to-face session. And concerning the etiological classification of hemolytic anemias, it may result from defects in the red cells, in the red cell membrane. The most common example is hereditary spherocytosis, which we are going to discuss later on. Defects in the enzymes in the red cells, whether they are energy potential defects. The most common and important is pyruvate kinase, but the most important is defect in the reduction potential defects. The protective enzymes, they protect from oxidizing agents, and the most common and important is G6PD deficiency, or hemoglobin defects, whether it is a disorder in the heme and this results in a disease called congenital erythropoietic porphyria. But the most important for us is defects in the globin chains, which could be quantitative or qualitative. If it is quantitative, we get thalassemia. If it is qualitative, you may get disease like sickle cell disease or hemoglobin C disease and others. Also, hemolytic anemias can result from causes outside the red cells, which could be related to immune processes or non-immune processes. Immune processes could, the best examples are isoimmune, means that the antibodies are naturally present, like hemolytic disease of the newborn and incompatible blood transfusion. I believe this will be discussed in the proper sessions. What we are caring about is autoimmune, the presence of autoantibodies, and these autoabnormal antibodies against self-antigens causing red cell destruction, which could be idiopathic or secondary to infection, drugs, hematological disorders like leukemia, immunological disorders like lupus and rheumatoid, or secondary to tumors. But there are non-immune factors causing hemolysis outside of the red cells: idiopathic, infection, drugs, what we call microangiopathic hemolytic anemia. These are conditions associated with microthrombi occluding the blood vessels. When they occlude the blood vessels, whenever the red cells they will pass, they get destroyed. It's hemolysis inside the small blood vessels. And there are miscellaneous conditions. The most famous of them is hypersplenism. In the next module, hemolytic anemia part 2, we are going to discuss the hemoglobinopathies. Thank you. Waiting for your questions.